top of your table, it says very turbid, somewhat turbid, barely turbid, not turbid. So y'all can make some notes about turbidity there, and then make a note if you were seeing any of your serratia, so like a red tint in your broth. The streak plate is the other one. Do y'all remember what the purpose of the streak plate was?
To single out the bacteria, to find the like
so
the
to dilute them out, and so we could isolate just one microbe. So on your streak plates, that very first quadrant that you streaked probably has a whole bunch of growth on it, and then you should hopefully have less and less growth as you move towards that fourth quadrant. So what it'll do is, if you look real careful at the colonies, some of them look different. I know on these plates that I've got up here, I can see several different bacteria. Some of the colonies are white, some of them are red, some of them are yellow, some of them are very defined small circles, some of them have really spread out and have taken a shape. So the shape that the colonies take, their color, whether they grow upward versus outward, tells you something about that microbe. And so if you look real close, you can pick out some differences in the colonies, meaning you might have more than one bacteria on there. And that's not possible when they all grow on top of each other in that first quadrant. But as you spread them across those other three, they get more room, and you can really see what they do when they grow. The deep tube, you should have made 3 stabs in the deep tube with an inoculating needle. And so if you look into the agar, you hopefully see 3 lines of growth down where you did those stabs. Hopefully it's a red line. Probably not though, right? Probably not. It's probably a white line is what you're seeing. You should see 3 of them. And then on the slant, you just inoculated the top of the slant. And so hopefully you're seeing some growth in your zigzag pattern. And so all I'm really looking for on this table is y'all to make some notes. One about, did you have your serratia, and did you have anything else? So just make some notes about what you saw. When you're writing your lab report for this one, think of the goal. The goal of this lab was aseptic technique with our serratia. So when you're doing your discussion, that's kind of the viewpoint you need to take when you're writing your lab report is aseptic technique was not maintained, and aseptic technique was not maintained. There was other microbes present in addition to serratia. If you had your serratia, you didn't have your serratia, then you say that you did not have your serratia. You definitely put your serratia on there. So why might not it have grown? We have not talked about it yet, because you may have a guess.
The other microbes of health?
Yeah, they compete with each other. And they'll do that with us too. So as a kid, you may have had this, or if you have kids, you may have experienced this. But when you have to take an antibiotic, one of the things that can happen if you have to take a very powerful antibiotic or take one for multiple rounds is you can get thrush in your mouth. Thrush is a yeast infection basically in your mouth. It's fungus. You have yeast and bacteria both in your mouth. And the reason that they don't get out of control is they compete with each other and they keep each other under control. But if you take an antibiotic that kills off all the good bacteria in your mouth, what happens to the yeast? Free range, right? Free range. And they take over. And so the same thing happens with bacteria. There's bacteria in the environment and they keep each other in check. In our bodies as well, we have good bacteria and good microbes. And so if you take a drug or an antibiotic that messes up the good stuff, you allow the bad stuff to set up. So you know, work in a clinic, C. diff, right? Clostridium differential. C. diff is a bacteria. And it's one that you have, but when you kill off all your good microbes, that one kind of takes over. And when it takes over, it makes people really sick. So yes, that is exactly right. Your serratia, you did put it on there, but it's not a real dominant microbe, and so it it was kind of overpowered by some of the other stuff that was picked up. Because this stuff that's in here, how often does it get sprayed with things and it's still here, right?
I sprayed it though.
I know, but how many years has this been a micro lab and how many years have those microbes been sprayed? Alright, they're here for the long haul. And if you contaminated your sample, you probably contaminated it with something like that that has been in here for a while. So they've got some arsenal to work off of. So just make some notes. There's no right or wrong way to make your notes. Just tell me honestly what it is that you're seeing on your stuff. And then that's what will go in your lab report there. Okay. Y'all can make your notes once we get started, because you're going to have a period here where you're going to have to sit and wait for a few minutes on this. So there will be a gap there where y'all can work on some of this stuff. This is all of the microbes that we're going to work with for this lab. This is actually a two-part lab where this is our staining lab. All the staining stuff we've been talking about, how to do a gram stain, so if you have gram positive or gram negative, how to do mycolic acid stain, how to do a endospore stain. Those are all the staining techniques that we're going to do next week. In order to do those staining techniques, we have to have microbes put on the slide and ready to be stained. And so that in itself takes a whole lab to do. Look at all the microbes that we've got to do here and all the different staining techniques we're going to apply. So here's your list. Let's kind of talk about these microbes for a second. We finished my table. I did not finish it. This will make more sense once I get to it. Gram staining, acid-fast staining, and endospores. Okay, so these are not in any particular order, just the order that came to me as I was writing them down. Bacillus cereus is one we have not worked with yet. So Bacillus cereus is the one that we find just on tabletops in general, and it is a common culprit of food poisoning. There's really not a lot of data on it, because how many of us, we get a little sick to our stomach, we don't run straight to a hospital or a clinic, right? We throw up a few times that night, we're fine the next morning, and it's not something that gets reported. So how often some of this occurs is a little hard to tell. Bacillus cereus is a gram-positive endospore forming. So it's gram-positive and it will form an endospore. So this is the same genus as Bacillus anthracis, so your anthrax. There's several that fall under this. So this one, when you do your gram staining, should test positive for an endospore, should also test as gram-positive. Yes?
I have to go, but I sent you an email. What do I need to do with them?
Just leave it there, I'll get it.
Okay, thank you.
Bacillus cereus does not form a mycolic acid layer, so it will be acid-fast negative. Does not form a mycolic acid layer. Mycobacterium flea. So what are you already maybe thinking out about this one?
Gram-positive.
Gram-positive. What else? Mycobacterium, mycolic acid. Yeah? Yeah? So y'all already know something here. Yes, Mycobacterium flea is a gram-positive. We know that because in order for it to form the mycolic acid layer, it has to be gram-positive. All mycobacteriums are gram-positive. So it is acid-fast positive, and it does not form an endospore. Mycobacterium flea is a bacteria that's not really associated with any type of human disease. It is actually found on Timothy grass out in the yard, and so it's not anything that's typically harmful to us. I use it because it's a safe one to study for the mycolic acid layer. Staphylococcus aureus is a very common bacteria. Can also be very dangerous when it's given the opportunity. It's a gram-positive. acid-fast negative, endospore negative, does not form an endospore. But this is when you have somebody who has a staph infection, this is what they're talking about, Staphylococcus aureus. Now, I left my projector on, I want to show y'all something here. Staph is something we have on our skins. They've done studies, babies born, 3 hours after they're born, they start to get colonized with staph on their skin. It's just part of what our good bacteria is on our skin. Feeds off the oils and things like that. But if you get a cut and you don't take care of it, what will that staph bacteria do? We'll take advantage of that opportunity. So they're opportunistic pathogens is what we call them, where normally they're fine. But if you give them the opportunity, then they will be a problem. What makes them such a problem, and we're going to look at this later on, but it's interesting now that we're working with it to kind of have it a little here. Being able to click on Google Image. Staph has a table that we can look at that shows all of the toxins that it can produce, and it's a very long table. Your book as well. Oh goodness. Well, anyway, I'll show it to y'all later on. I didn't have it up, but staph has most of these bacteria that make us sick. They have like 1 or 2 toxins, other enzymes that they can produce that either make a blood clot or lyse our red blood cells or something like that. Just 1 or 2 things. Staph has a list of about 13 things that it can produce that make us sick. Everything from being able to break down the antibiotic to lysing our red blood cells, making our blood clot, producing these endotoxins and exotoxins that we're severely allergic to that cause toxic shock syndrome. It's got a list of about 13 things. Destroying white blood cells. It's very unusual. Other microbes don't do that, right? They got 1 or 2 things, and Staph's got this huge arsenal of stuff that do all these really bad things for us. Now, normally it's fine, right? But if it gets somewhere it's not supposed to be, and it starts doing that, then our body reacts to that. So we'll look at that later on. But again, the staph that we use in here has been genetically disarmed. We're always careful, but it's been genetically disarmed. Shigella flexneri. This one is, most of the time when it's talked about, it's just called Shigella. It causes a very severe form of diarrhea, especially in kids. It's a gram-negative, so it's one of the gut microbes. The most common way that it's picked up is is a fecal contaminant. So daycares, kids, right? It's gram-negative, and it's negative for all of these things as well. But it's a fecal coliform, and it gets picked up, and if it gets oral contamination, it will go down in the GI tract, and it causes really severe cramping, really severe diarrhea, bloody diarrhea. It'll usually clear up on its own in about a week, but you're very miserable while that's happening. Very common in kids because they just touch everything and then touch their mouths. Daycare workers changing diapers and stuff, sometimes they'll get this as well. E. coli, this one is a gram-negative, so Escherichia coli is also negative for all of these things. Another fecal coliform, and again, you're supposed to have it as long as it stays where it's supposed to be, it's fine. But if you get a fecal contamination and pick it up in some way, whether it's through animals being processed or it's maybe E. coli in their gut, and if you eat deer meat where someone nicked the gut or something while they were cleaning the meat, Same way with beef in a slaughterhouse or poultry in a slaughterhouse. If the gut contaminants are not taken and discarded carefully in a correct manner before the meat is exposed, you can get some E. coli contaminants on the meat. And then that's where you'll hear on the news, oh, we've got some contaminated meat, or we've got some this, and you get a little sick from it. Normally, if you're a healthy, just regular, things functioning like it should, you get a little sick and you're fine. But what if you're immunocompromised, or you're a young child, or you're an elderly? If it's somewhere it's not supposed to be, then it can definitely be a problem. Serratia, we've already worked with. Yes, do you have a question? No. Serratia, we've already worked with. What do y'all remember about Serratia? Gram-positive or gram-negative? You got 2 guesses.
Remember.
It's gram-negative, right? Your Serratia is gram-negative. Pigment's red. It's actually the same broth from last week. We already had it made up, so we just kept it alive for this week, and we'll use it again. So it should test negative for all of these things here. And then there's an unknown, and Mr. Holt made them. I have no idea what he put in the unknown, so we're just going to test it, and I'll ask him later. And we'll see what it tests positive and negative for, and see if we can guess what it is. But these are all your microbes that you've got for this week. Now, these are the 3 staining techniques we're going to be doing. So we have a staining procedure where we will take all of these microbes and we will perform a gram stain on all of them. And I've already told you what's supposed to happen, but y'all need to see if you can do it correctly and see if these are results that you're getting. Acid fast, we have a staining procedure that identifies if our microbe has a mycolic acid layer. It's a waxy layer. So if they have it, then they're acid fast positive. If they don't, then they're acid fast negative. Again, we already know what's supposed to happen. Let's see if we can get that to happen. And then we have a 3rd staining procedure that we'll do next week for the endospore, where you can determine not if your microbe can form an endospore, but if it currently is an endospore. This will show you the difference in an endospore and a vegetative cell. And so again, we already know what it's supposed to look like there. What this means is you'll have a lot of slides to make today, because you're going to need to take your Bacillus cereus and make 3 different slides of Bacillus cereus. You've got to make 3 different slides of your Influi, 3 different slides of Staph aureus, 3 different slides of Shigella, so on and so forth. So you've got 1, 2, 3, 4, 5, 6, 7. You got 21 slides that you've got to make, and then we're going to stain them all next week. This is one where you are welcome to work by yourself, but I highly recommend that y'all work together in some way, pair up something, because this is a lot to do for just one person. And y'all can share slides between each other once we get them all made and everybody look at something. But staining next week, I definitely want everybody to do some of the staining, and I'd like for all of y'all to make up some kind of slide so that you know how to do that as well. But y'all are welcome to work together on this one. Labeling is going to be your best friend, because once you get that microbe on the slide, you're not going to have a clue what's on there unless you've labeled it. So once you've got your stuff in the Copeland jars, but before you ever start pulling anything out, I would get the tape and I would get all my labels ready. So you would need to label what microbe you have, and you can abbreviate. So like Bacillus cereus, you could just write BC. Staph aureus, SA. But you need to write like BC gram, BC AF for acid fast, BC spore for endospore. So you need to label what microbe is going to go on that slide and what staining procedure is going to be applied to that slide. And just a little tiny, as small as you can get it on there, just enough to have a label. Right, just a little piece of tape. And see if you can write that on there, just however big you need to write it. And get all of your labels made up, have them set out at the edge of a table, and then when you pull your slides out, you can go ahead and label them all. And then start applying your microbes to them once you get them all labeled. Probably the best way to do this is going to be to pull out a huge sheet of paper towel here and lay it out. And then as you pull your slides out of the Copeland jars, start lining the slides up, right in pairs or so, or however. Get them all laid out, label everything, and then get your microbe and start applying the microbes to them. how to apply the microbe to your slides. So there's a place in your lab manual that tells you how to do that. Again, I lost my tab one, so I don't know what page this is on. Pull up our lab manual. Okay, it's on page 178. Okay, page 178. There is a pictured procedure, 3.41 procedural diagram making a bacterial smear. So page 178. There is one modification I need y'all to make. It says on the very first step, step 1 in the picture, wearing gloves and eye protection, place a small drop of water on a clean slide using an inoculating lube. We are already working with a broth, so you do not need to put water on there. We already have a liquid that we're working with, so you do not have to do step 1. And we also don't wear eye protection in here. I mean, I guess you could if you wanted. We don't wear eye protection, but you do need to glove up. Step 2, aseptically add bacteria to the water. You're just going to add your broth to the slide. So you're going to take a disposable inoculating lube. Find it, Brittany?
Yeah.
You're going to take a disposable inoculating lube, the yellow ones, and you're just going to get a very tiny amount. So those loops are designed to pick up a very specific amount of broth. So just get whatever that loop picks up and tap it down on the slide and then spread it around. One of the things that y'all have to do is you have to let the slide dry. So if you put too much on there, you're going to be here forever, right? Very tiny amount. That tiny amount is going to have thousands of microbes in it. So very tiny amount, spread it out. Don't blow on it, right? What does that do? Do not blow on it. We're wearing masks, so that shouldn't be a problem this semester. You can, if you need to, fan it or wave it around. I know that seems counterintuitive, but that's going to happen regardless, stuff falling on there. Practice your aseptic technique, but know that you're probably going to have other stuff on there just because of the way we're set up. I want y'all to just put one emulsion on every slide. It says that you can put 2 or 3. Just do 1 per slide because we're going to do different staining techniques on them. Let's see. Step 3, allow the smear to air dry. Yes, we did that. Step 4, use a slide holder, pass the smear through the upper part of a flame. So you got to flame it. It's called heat fixing them. So you're going to light the Bunsen burners. Again, right? Carefully. Light the Bunsen burners. And then you've got some forceps in your kits. You also have some test tube holders, however you want to hold the slide. But once you get your flame going, you're going to take that slide, holding it with the holder, and you're just going to wave it across your flame 2 to 3 times. What you're trying to do with the heat fix, it does 2 things. One is it helps your cells adhere to the microscope slide. They're supposed to do that anyways, and this helps. It only works if your sample was dry. What it's doing is it's dehydrating a little bit in between the cell and the slide. The cells themselves have a negative charge to them, and your microscope slides have a positive charge. So they're naturally going to be attracted to each other, but you got to dry that layer between them and let them stick. This helps with that when you flame it. If you don't flame them correctly when we do our staining, they might get rinsed off. So flame them, let them dry is the big thing, and they should flame correctly. Letting them dry. I know that's hard because that's the patient part. We just got to sit there. Don't put very much on there, and it'll take them real long to dry. But once they're dry, you can start flaming them. Once you get them flamed, we're all done. How you'll store these, and you can see other classes have already done it. You'll probably have to get a fresh one because you'll have to put alcohol over this one from your Copeland jars. But you'll get a fresh paper towel. You'll line the slides up just like you had them, but you'll roll them in the paper towel. So I've got 2 slides here. I've rolled them up. I've got 2 slides here. I've rolled them. 2 slides. Roll them. Just roll them up in the paper towel, and then we'll put some tape on them. Put your name on there, whoever's name, and then we have a bowl where they'll go, and then they'll be ready for next So, the staining part next week is not difficult, but it's very frustrating to do all of that work and not have a microbe on there. So, take your time today, get your microbe on there, let it dry, heat fix it, do all this stuff right today, and you'll have the microbe next week for the staining, because that's frustrating when it's not there. And let me know, I'll help you out if you have any questions, okay? All right, so, everybody good? I'm trying to think here. I haven't pulled the beakers out yet, 'cause I'm trying to keep them as aseptic as possible. I'm fixing to get the beakers out. But you've got all the broths here where y'all can pour some. Y'all are the very last class. It's not always the best thing because you're at the mercy of how careful the other classes were. Hopefully they were careful. But you can get some beakers, get them labeled, use the parafilm, keep everything as covered as you can. And I think they forgot we had lab today because they cleaned everything. We had a waste jar here. I'll get it back out and get our dirty beaker stuff set out. So when you're done with all of your microbes, you pour them in the waste jar, put your dirty beakers in the dirty beaker container that I'll have here. And then any trash that you have, I'll give you all your trash bags for the table. When you're done, we'll put them up here in these trash bins, just like always. When y'all are finished with your stuff from last week, just dump it straight into the trash cans when you're finished with it. So unless you have a reason you want to keep it, you're welcome to just dump it. Any questions? Okay, I'll get all this out and y'all go